Please listen carefully.
Hello and welcome to GasGasGas, the podcast that covers the FRCA primary exam. We're going to fit into your day and give you as much of your life back as you could possibly imagine. I'm here to make your studying easier. Listen to us on your commute, in the gym, in the shower, or when you're ironing your scrubs. Expect facts, concepts, model answers and the odd tangent. Check out the show notes for all the detail and remember to follow the show so that you never miss an episode. Let's get on with it.
3, 2, 1, and we are on air with GasGasGas, and here we are today with Tom, willing victim, returns for yet more punishment. Tom, I hope you've set the sensitivity on your microphone high enough that we can hear the sweat bead on your forehead.
I mean, I really hope I haven't set it that high, but you know, if I have, and that's the kind of thing you enjoy, best of luck to you.
It certainly warms my heart. Right, so we're going to pile straight in to your pharmacology Viva. Are you ready for action?
Yeah, let's go.
Lovely. So, Tom, I'm going to set a timer because we're trying to be better about this today, aren't we? What do you mean by the term pharmacokinetics?
So, pharmacokinetics encompass all of the things that your body does to a drug. So it's normally broken down into absorption, into distribution, metabolism, and excretion.
Okay, lovely. And could you just break those four things down a little bit for me, please?
Sure. So when a drug is taken, if it's not taken intravenously, you have to first consider how it's going to be absorbed. So some drugs may be absorbed well through the stomach, some not so well, may be absorbed in the intestine. If it's given via a parenteral route, then absorption is less of an issue. Drugs can also undergo first-pass metabolism, so if they are absorbed in the gut and transported via the portal system to the liver, they may be metabolized and a significant proportion of them may be removed from the circulation before they have a chance to have activity. So all of those things need to be taken into account when administering a drug. In terms of distribution, so different drugs may have different affinity for different tissues. So drugs that are easily soluble in lipids will travel quite freely across cell membranes and into lots of distant parts of the body, whereas more water-soluble drugs may be retained more within the vascular compartments and reach their target sites more slowly. So that also needs to be taken into account when administering drugs. Following their distribution, they are usually metabolized before excretion. Some drugs are excreted unchanged, but the reactions that modify them into forms that are either inactive or excretable will be important for how they act and how long they act for. Whether their metabolites are active or toxic, again, is also going to be an important consideration when administering drugs. And then finally, excretion, whether they're excreted via the kidneys, via the bowel, whether they're exhaled, it's going to be important in deciding how a drug is used and in which patients it's likely to accumulate or have, slightly different or prolonged effects.
Okay, lovely. You mentioned bioavailability. What factors might influence the bioavailability of a drug?
So, bioavailability is going to be influenced first by the route via which a drug is given. If a drug is given intravenously, it will be 100% bioavailable. That means that it will circulate via the heart into the systemic circulation without being significantly metabolized by the liver. If it's taken enterally, however, then it may be, if it's absorbed in the bowel, it will go via the portal venous system directly to the liver. Now, if a drug is significantly metabolized into some inactive form by the liver, then it will have significantly reduced effect when given orally compared to when it's given intravenously. And so the percentage of the drug that's removed in that initial pass through the liver refers to its first pass metabolism and dictates what percentage of a drug is bioavailable. And if only 50% of a drug makes it past the liver, having been absorbed in the gut, then it's only 50% bioavailable. An example of a highly bioavailable drug would be paracetamol. It's well absorbed and undergoes very little first-pass metabolism, whereas a drug such as vancomycin would be at the opposite end of the spectrum, and rather it doesn't actually involve metabolism in the liver, but it's not absorbed at all, so its bioavailability is zero if taken enterally.
Cool. Any other factors that might affect how that liver metabolizes a drug? The rate it works at, for example.
The rate it works at. So certainly some enzymes that break down drugs can be activated or inactivated by other drugs in the system. So if you take...
And that is your 5 minutes, Tom. Lovely. We're going to move on a bit there. What drugs are used to reduce blood sugar in diabetes mellitus?
So medications used to reduce blood sugar in diabetes can be broken down into their modes of activity. So there are medications that act directly on the liver and peripheral tissues. This includes insulin. There are medications such as metformin that increase the body's susceptibility to the effects of insulin or its insulin sensitivity. There are incretins, which affect the body's insulin secretion as well as controlling appetite and essentially intake of dietary sources of sugar. And there are medications that act on the renal system, such as the flozins, which promote excretion of glucose.
Okay, lovely. Just focusing in a little bit more. Tell me about insulin.
So insulin is a hormone produced in the beta cells of the pancreas, and its primary function is to control peripheral absorption of glucose into various tissues, but including skeletal muscle, the liver, and other peripheral cells, including in the central nervous system. It acts via binding to tyrosine kinase coupled receptors on the surface of cells, resulting in downstream signaling that promotes the uptake of glucose into the cells where it can be used as an energy source.
Mm-hmm. And what various insulin preparations are you aware of, broadly?
So insulin preparations are divided into long-acting, short-acting and rapid-acting insulins. Long-acting insulins, as they sound, act for long periods of time and give, particularly in people with type 1 diabetes, give a basal level of insulin so that the body doesn't suffer from ill effects of insulin's complete absence, such as hypoglycemia or diabetic ketoacidosis. To then cope with dietary intake and spikes of glucose, you can give shorter acting insulins that try and coincide their peak effect with intake of dietary sources of glucose. So Novorapid would be a typical short-acting insulin that allows you to do this. And there are intermediate-acting insulins as well, which can be used as part of insulin regimes to try and place insulin in the body when it's in demand for particular patients based on their patterns of glucose spikes or plasma glucose spikes.
And which medicines, classically for type 2 diabetes, are associated with hypoglycemia?
So glucoside and insulin specifically are associated with hypoglycemia. Insulin, as its primary action, is to reduce plasma concentration of insulin. Glucoside, because it promotes the action of insulin itself and therefore causes a reduction in blood glucose.
Okay, lovely. And do you know of any other medicines for type 2 diabetes at all?
That cause hypoglycemia or...
No, just for general treatment.
So we've spoken about glucosides, metformin, insulin, the flozins, And there are new medications now that have been used for weight loss as well as for treatment of insulin, which again are incretin hormones that affect appetite, such as semaglutide and liraglutide. Yeah.
And how does metformin differ to glycoside?
Metformin differs to glycoside in that rather than promoting the secretion of insulin, it sensitizes peripheral tissues to the action of insulin and therefore doesn't produce hypoglycemia.
Does it have any implications for unwell patients?
Yeah, so one well-known side effect of metformin can be lactic acidosis, which is in a mild form in a well patient may be of no clinical significance, but in a critically unwell patient, worsening acidosis and lactatemia could have significant negative effects.
Excellent. That is your 5 minutes there, Tom. Do you need a glass of water? Are you all right, mate?
No, I'm good.
Okay, lovely. So, what do you understand by a clinical trial?
A clinical trial is a research project in... Sorry, give me a moment to think about this because it's a definition I've not considered before. A clinical trial is a process by which medical interventions are tested via scientific method by systematically selecting patients and trying to test the effect of interventions on that group, which can be done in various different ways, whether it's through randomized controlled trials or... Sorry, go on.
I was going to ask you, what types of clinical trials are you aware of?
So, often considered the gold standard for a clinical trial would be a randomized controlled trial. This is a design of clinical trial whereby an intervention is tested against pre-decided outcomes. So, specifically, there will be a control group in which no intervention is given. Often the participants and sometimes the administrators of the intervention will be blinded to whether or not participants are given the intervention, and then that will be compared against... Yep.
Sorry, Tom, are there any other types of clinical trial you're aware of?
Yes, so there can be prospective studies and retrospective studies that look at the effects an intervention had and attempt to control for confounders using techniques like regression analysis in order to tease out whether or not there is a true effect on the population of interest.
And you mentioned randomized control trials. If a pharmaceutical company is taking a drug to market, how might they approach that?
So pharmaceutical companies have to do phase 1, phase 2 and phase 3 studies in a clinical trial. So phase 1 clinical trial will be a safety study initially, which will be on a small group of people and will establish that a medication is safe to administer and safe to use, broadly speaking. Phase 2 trial will begin looking at whether it has a specific effect as an intervention. For instance, whether it reduces the rate of heart attacks in a particular population. So a certain number of people that will be tested, which will be dictated by statistical analysis prior to the trial beginning. And following this, phase 3 study will look in much larger numbers after a medication is used publicly to see whether the initial findings of the phase 2 trial still hold true and to find out any more rare adverse effects that may be important.
How might you analyse multiple trials to identify a positive outcome for a particular intervention?
So in the absence of a well-designed randomised controlled trial to show whether or not an intervention is effective, it can be possible to combine the results of multiple trials looking at the same intervention through something called a meta-analysis, meta just meaning after, so an analysis after the fact. This involves finding as many trials as are relevant, carefully analysing each of them to see whether or not they were performed effectively, whether or not the outcomes they're looking at were the same or comparable, and whether or not the populations themselves were comparable and whether the intervention was actually in fact the same. If all of those things are true and they can be grouped together, then a scatter plot can be used to try and show with a range of uncertainty whether or not all the trials taken together show a positive, neutral or negative effect on the outcomes of interest.
Lovely. I'm going to stop you there. That is your 5 minutes, 3 5-minute blocks, 15-minute station.
That went fast.
Yeah, because we were meandering a lot with the other ones we've done. I just want to point out, Tom, just for clarity, I think I've got down here a phase 1 trial, small group, make sure that it's not lethal, and they give a range of doses. It says here
dose finding and safety, yeah.
Yeah, and then phase 2 is different regimens to ascertain potency. Neither 1 or 2 are randomized. And then 3 is you've figured out the optimal dose, you've figured out the optimal frequency, and then you do a proper randomized controlled trial. And then 4 is that post-market surveillance, yeah.
I forgot there was a 4, and so I got myself muddled. Yeah, I've not looked in a while. Thanks for the clarification.
No, you did well to do that. I thought that was a humdinger, so well done. And you took it with grace as well. You didn't just you just kept the on the inside quite well after your brief. How the hell do I define what a clinical trial is moment? And that's the right move.
Yeah, okay. It's good to practice as well, though, because I can... I'm in the stages at the moment of practicing saying things out loud and making sure that I've got at least an opener to lots of the things that come up. So there's another one for me to practice.
Yeah. Hang on, let me think. Okay. I'm going to stop it there then, Tom.
Thanks for listening to that episode, guys. If you found it useful or awful, please like and subscribe and rate the show. Definitely go check out those show notes on GasGasGas.uk. We all know that this is a bucket of content. I want you to take some time for yourself and don't overcook it. Don't freak out. Keep studying.